To kick things off, we need to answer a fundamental question. What are organoids? Simply put, organoids are 3D lab-grown miniatures of human organs. They aren't just a random collection of cells in a petri dish. They are organ-like structures that mimic the actual architecture, genetic makeup, and biological functions of everything from a human brain to the liver, heart, and lungs. For decades, medical research relied on two things: 2D cell shock cultures, which are too simple, and animal testing, which often fail because, frankly, a mouse is not a human. Organoids change the game by providing a human-relevant model. They are grown for stem cells, either embryonic or reprogrammed adult cells, that have the incredible ability to self-organize. When given the right environment, these cells know how to build a kidney or a gut on a microscopic scale. But why does this matter? Because organoids allow us to practice personalized medicine. We can take a patient's own cells, grow a mini-me of their specific organ, and test which cancer drugs work best on their unique biology before they ever take a single pill. They are also revolutionizing drug discovery and our own understanding on how certain viruses works, like COVID-19 or Zika, which attack human tissue. However, a functional mini organ in a lab isn't as simple as putting cells in a jar. It requires a highly controlled, engineered environment to thrive. To explain the physical infrastructure needed to make all this happen, I'll hand it over to the member 2 to discuss these bioprocess fundamentals.
Bioprocess fundamentals are essential for the development of organoids, which are advanced three-dimensional in vitro cell culture systems that recapitulate the architecture and functionality of native tissues. The three critical components in this process are scaffolds, bioreactors, and culture media. First, scaffolds provide a biomimetic extracellular matrix environment that supports cell adhesion, proliferation, and differentiation. Materials such as hydrogels, collagen, alginate, and matrigel are widely used due to their biocompatibility, porosity, and tunable mechanical properties. These scaffolds facilitate cell-to-cell and cell-to-matrix interactions, which are essential for self-organization and morphogenesis in organoid systems. Next, bioreactors play a key role in maintaining controlled physicochemical conditions. They enable precise regulation of parameters such as pH, temperature, dissolved oxygen, and shear stress. Advanced systems such as stirred tank bioreactors, spinner flasks, and perfusion bioreactors enhance mass transfer, nutrient diffusion, and waste removal. This improves scalability, reproducibility, and viability of organoids, especially during long-term culture. Finally, culture media provides the necessary nutrients and biochemical cues required for growth and lineage-specific differentiation. It typically contains amino acids, vitamins, glucose, growth factors, cytokines, and signaling molecules such as WNT, BMP, and FGF pathways regulators. The use of serum-free and chemically defined media ensures better standardization and reduced variability. In conclusion, scaffolds offer structural and biochemical support, bioreactors ensure optimal bioprocess control, and culture media drives cellular signaling and differentiation. Together, these elements enable the efficient production of organoids for applications in disease modeling, drug screening, personalized medicine, and regenerative engineering.
When we focus about organoids, we often focus on the magic of stem cells, how they self-organize into mini brains or kidneys. But the real unsung hero is process control. In a human body, our heart, lungs, and kidneys work 24/7 to maintain a perfect internal environment. In a lab, we have to build that biological autopilot from scratch. Today, let's look at how we control oxygen, pH, and nutrients to keep these tissues alive. First, let's talk about oxygen and pH. In 2D cultures, cells just sit in a thin layer of liquid, but organoids are 3D spheres. This creates a massive problem, the necrotic core. Without precise control, oxygen can't reach the center, and the organoids die from the inside out. We solve this using perfused bioreactors, essentially mechanical hearts that keep the fluid moving so oxygen stays at physiological levels. Closely tied to this is pH control. As these mini organs eat, they produce metabolic waste like lactic acid. If the pH drops even slightly below 7.2, the cells stress out and stop specializing. We use automated CO2 injection systems to buffer the media, keeping the acidity exactly where it would be in your own bloodstream. Then we have nutrient and growth factory delivery. This isn't just about feeding the cells, it's about signaling. To grow a complex tissue, you have to change the menu at specific times. On day 5, you might need a spike in glucose. On day 10, you need a specific protein to tell the cells to form a blood vessel. Process control allows us to automate this feeding schedule. By using microfluidics, we can deliver microscopic drops of nutrients at the exact millisecond they are needed, ensuring the organoid develops into a structured organ rather than a random clump of cells. Finally, none of this works without monitoring. We can't just hope the levels are right. We now use integrated biosensors, tiny electrodes inside the growth chamber, that gives us a live dashboard of the organoid's health. If the sensors detect a dip in oxygen or a spike in waste, the system adjusts the flow rates instantly. It's a closed loop system that acts as an external nervous system for the tissue.
Now I will talk about the scale-up challenges and applications of organoids from the bioprocess engineering perspective. Although organoids are successfully grown in laboratories, scaling them up for large-scale production is still a major challenge. One of the main problems is maintaining uniform growth. When organoids increase in size, cells at the center do not receive enough oxygen and nutrients, leading to the cell death. Another major issue is the lack of vascularization, meaning organoids do not have blood vessels. This limits nutrient transport and restricts their size and functionality. In addition, controlling parameters such as oxygen transfer, nutrient supply, and waste removal becomes very difficult during large-scale production in bioreactors. This makes the process expensive and less efficient. Despite these challenges, organoids have important applications. They are widely used in drug testing, where they provide more accurate results compared to animal models. In cancer research, tumor organoids are used to test patient-specific treatments, which is a step towards personalized medicines. In future, organoids may also be used for organ transplantation, but this requires further advancement in bioprocess engineering and scalability. To conclude, organoids have great potential in modern biotechnology, but their successful application depends on overcoming scale-up challenges. Bioprocess engineering plays a crucial role in transforming organoids from small laboratory models into practical medical solutions.
Thank you for watching the video. We hope you like it.